here we have the axon, and this axon is myelinated. Okay, so myelination provides insulation. So it turns out that there are no voltage-gated ion channels in the myelinated region. All the voltage-gated ion channels are actually localized to these nodes of Ranvier, which are in the gaps between the myelinated region. So what this allows to happen is it allows for the action potential to be jumping from one node to the next node. So it starts with the axonal hillock, jumps to the first node, the second node, third node, etc. Okay, so the action potential used to be here, right? The sodium would be rushing in, right? And then those sodium ions are going to go down to the next node on Ranvier, and then basically trigger another action potential by opening up all those voltage-gated sodium ion channels, right? And then after the voltage-gated sodium ion channels in this node close, the potassium voltage-gated ion channels are going to open. Potassium is going to rush out, right? And then we're calling this the red is showing the repolarization. The blue is showing the depolarization, right? So red means potassium's rushing out, and the blue means the sodium is rushing in. So what's going to happen is this is now the action potential is now going to jump from this node to the next node to the next node, etc. And that's how it's going to travel very rapidly down a myelinated neuron. For this table, what I want to just have you take from this is if you look at the diameter of a nerve, the thin nerves tend to have very slow velocity of conduction, okay? And very high diameter nerves have a very high velocity of conduction. So this is for a very thick 12 to 22 micrometer diameter nerve. We see the conduction is about 70 to 120 meters per second. Huge, right? However, in a very thin nerve, it's 0.3 to 1.3 micrometers diameter. We see 0.7 to 2.2 meters per second of velocity of this, right? So you don't have to memorize these numbers or the examples over here. What I just want to point out is that the thicker the diameter of a nerve, the faster they can conduct the traveling of an action potential down the nerve, and remember in this case in the top, these are definitely myelinated nerves that can be having such a high velocity of transmission or conduction. So the next thing I want to do is now discuss the synapse. So the synapse is the functional connection between a neuron and the cell that it's talking to, right? So in the central nervous system, the second cell is going to be another neuron typically, and in the peripheral nervous system, the second cell could be a muscle, organ, or gland. So one thing to talk about are electrical synapses, and I think I mentioned this in the previous lecture, but electrical synapses occur both in smooth muscle and cardiac muscle, and in also parts of the brain. And these kind of cells have gap junctions. And what gap junctions allow is the electrical stimulation to be passing from one cell to another cell in real time. So for example, in the heart, we need the atria and the ventricles to be contracting at the same time. Gap junctions between the heart cells allow them to all contract simultaneously. So we'll be talking about that again when we do the cardiovascular system. But I just want to point out, so these gap junctions, which are use the connexin protein really play an important role in the body and allow for all the cells in a tissue, for example, on the heart, to do the same thing at the same time. And these cells have to be the same resistance to be having to sharing electrical stimulation. So here I just want to show you, again, this picture I showed you before, but here's the cytoplasm of one cell, the cytoplasm of another cell, and they are connected by these connexin proteins that allow electrical signaling or stimulation to travel through both cells at the same time. So that's one kind of stimulation, right? The other thing I want to talk about now are chemical synapses. And when we think about the nervous system, we usually think about neurotransmitters and as the molecule nerves secrete. And basically the neurotransmitters were discovered by Otto Loewi, and he won the Nobel Prize in 1936 for this discovery. You don't have to memorize that, I'm just giving you a little piece of history. And he really was the one who discovered that nerves secrete neurotransmitters, and he actually studied how neurotransmitters affect the beating of a heart. And one important thing I want to introduce now is that where nerves secrete their neurotransmitters is called the synaptic cleft, and the presynaptic cell is the one that is going to secrete the neurotransmitter. The postsynaptic cell is the one that's going to receive the neurotransmitter, and basically the presynaptic nerve and the postsynaptic cell basically are held together by the cell adhesion molecules called CAMs, and they really help to make a tight interaction between the two cells in the synapse. So here is, we call it the terminal button of an axon, right? And here are all these vesicles that have neurotransmitters, and here is the a cleft, right? So basically the neurotransmitters can be released into this synaptic cleft, and then the cell down here can basically receive the neurotransmitters and then make an appropriate response. So in this case, the receiving cell is a skeletal cell, and maybe this neurotransmitter is telling it to go and contract, for example. So how are neurotransmitters released by an action potential? So what happens is when an action potential gets down to the bottom, or the end of a nerve, which of axon, end of the axon, which we call a axonal terminal, the action potential will then, when it gets to the axonal terminal, will trigger some voltage-gated calcium channels to open. Okay, so 1, action potentials reach the axonal terminals. 2, this, remember, it changes the voltage, right? The resting member potential will be increased from the sodium, right? And then what happens is the voltage-gated calcium channels are going to open. Calcium is going to rush in. So in order for this to happen, right, we can assume and understand that there's a concentration gradient where there's more calcium outside the cell than in the inside the cell. Otherwise, when you open these channels, the calcium wouldn't be coming through. Okay? Calcium is going to bind to a special protein. Okay? And basically, this calcium complexing with this special protein called synaptogammon is going to lead to the exocytosis of these vesicles. So here's the vesicle containing neurotransmitter. Okay, the snare complex basically helps this vesicle attach to to near the plasma membrane, but it cannot exit and fuse with the membrane without calcium and this special combining with a special protein. We call that just a sensor protein, and it's going to make it's going to bind this protein and basically make this calcium protein complex that allows the release of this vesicle via the process of exocytosis. So I just want to summarize the neurotransmitter action. Okay, so we have the presynaptic neuron and a postsynaptic neuron. Okay, the presynaptic neuron is the one that's going to be releasing a neurotransmitter. It's going to have an action potential traveling down the axon. This is going to lead to the opening of voltage-gated calcium channels. Calcium will bind that special protein that helps vesicles perform exocytosis. That'll result in the release of neurotransmitter into the synapse. Then what will happen in the postsynaptic neuron is what's going to happen is there'll be chemically regulated ion channels that'll open. So basically the neurotransmitter is going to now trigger the opening of a chemically regulated ion channel. This will allow the influx of sodium. Okay, the concentration gradient of sodium is going to let it pass inside the cell. This is going to cause depolarization. We call this an excitatory postsynaptic potential, EPSP. What this will do is going to create a localized production of depolarization in the part of the recipient cell that is at the synapse. Okay, and of course, if enough neurotransmitters are released, the sodium can diffuse through the cell and perhaps have enough signal to get to the axonal hillock to trigger an action potential in the second nerve cell. And if it does, so if you have enough excitatory postsynaptic potential received, then it'll open the voltage-gated sodium ion channels, right? And then we know that once it hits the certain threshold, it'll then open the voltage-gated potassium channels, and then action potential will be transmitted down the axon, and then its axon terminus will then release neurotransmitters to stimulate the next cell, right? And then just to show you how the cartoon of this, right, you have the cell body, right, and some dendrites, and basically the presynaptic axon is going to be releasing neurotransmitter that this second nerve cell, the postsynaptic nerve cell, is going to pick up, right? These in the postsynaptic cell, there are going to be chemically, not voltage gated, chemically gated sodium ion channels that will open, and the neurotransmitter is going to open those chemically gated ion channels, so allow sodium to rush in. It's going to be something called the EPSP for excitatory postsynaptic potential. That will then increase the membrane potential in these cells so there's enough signal that the sodium ions can diffuse down to the axon hillock. If it hits the negative 60 membrane potential, it'll then trigger an action potential to travel down the axon. We can see that this is a myelinated axon. We can't tell if it's oligodendrocyte or Schwann cell in this image. We can just tell that it's the myelin sheath. We don't know what's causing that at this point. But the action potential can jump from the axonal hillock to the node of RMVA, to the second node, etc., and all the way down to the axonal terminal, where then it will then secrete neurotransmitter. And this concludes the first half of the lecture, and I will continue the second half of the Chapter 7 lecture in a couple days. Thank you for your attention, and have a great week.